Today we are going to discuss the structure of DNA. In this we will discuss what is a nucleotide, nucleoside, what is DNA double helix, what is complementarity, antiparallel orientation, hydrogen bonds, codon and central dogma. So let's see first what is a nucleotide. A nucleotide has three parts. One is the deoxyribose sugar, another a nitrogenous base, and third a phosphate group. Let's discuss in detail. The deoxyribose sugar has 5 carbons. Here it's the first, second, third, fourth and fifth carbon. To the first carbon, the nitrogenous base is attached, and to the fifth carbon, the phosphate group is attached. The phosphate group has a negative charge. That means it can give protons. That's why DNA is acidic in nature. Now coming to the nitrogenous bases. The nitrogenous bases are of two types. One are the purines, adenine and guanine. They are the purines. The other type of nitrogenous bases present in DNA are pyrimidines, thymine and cytosine. Thymine and cytosine, they are the pyrimidines. So let's see the bonding. The nitrogenous base is attached to the deoxyribose sugar by N-glycosidic linkage, and the phosphate group is attached to the deoxyribose sugar by a phosphoester bond. So there are two types of bonds in a nucleotide. One is a phosphoester bond and another is the N-glycosidic linkage. Now let's see what is a nucleoside. If we remove this phosphate group from the nucleotide, so what remains is that a deoxyribose sugar and the nitrogenous base. These two form the nucleoside, and the bonding present is only the N-glycosidic linkage. So this is all about a nucleotide and a nucleoside and purines and pyrimidines. Now let's see what is the DNA double helix. One molecule of DNA has two strands of DNA. They are coiled around each other. One turn of our DNA double helix model has 10 base pairs, and each turn has covers a distance of 3.4 nanometer. It is stabilized by hydrogen bonding. Now we will see what the two strands of DNA are complementary to each other. Complementary just means that adenine always pairs with thymine using 2 hydrogen bonds, and cytosine always pairs with guanine using 3 hydrogen bonds. Here also we can see thymine always pairs with adenine and guanine always pairs with cytosine. So if we know the base sequence of one strand of DNA, we can deduce the base sequence of another strand of DNA. This is the complementarity rule. Then coming to the antiparallel orientation, it means that the 5' end of the deoxyribose has a free phosphate group at one end, and at the other end The 3' end of the deoxyribose has a free hydroxyl group. So we see that the orientation of one strand of DNA is 5' to 3'. Let's see the other strand. Here at the top, it's the 3' end which is free, has a free hydroxyl group. Here was the 5' end which was free and it had a phosphate group. But here it's the 3' end which is free and it has a hydroxyl group. Similarly, going downwards, the 5' end is free and it has a phosphate group. So the orientation of this strand is 5' to 3' upwards, whereas the orientation of this strand is 5' 3' downwards. So this is upside down and this is downside up. So this is the antiparallel orientation of the two strands of DNA. And now coming to the stability of DNA. The DNA molecule is stabilized by hydrogen bondings and the base stacking interactions. Now let's see what is a codon. A triplet of the consecutive bases present in the triplet, that is adenine, thymine and cytosine, here form a codon, and one codon is responsible for one amino acid when the DNA is finally replicated and then translated into transcribed. When our DNA molecule is transcribed to RNA and then translated into DNA, one codon of DNA would be responsible for one amino acid. And this is also the central dogma, that is that DNA first transcribes into mRNA and then mRNA then translates into proteins. So today we have covered all the topics. We have discussed what is a nucleotide. A nucleotide is formed of three parts, the sugar, nitrogenous base and the phosphate group. And if we remove the phosphate group, what remains is the nucleoside. Then we discuss the DNA double helix model, and then we discussed what is complementarity rule in DNA, that is adenine always pairs with thymine and cytosine always pairs with guanine. Then we discussed the antiparallel orientation of the two strands of DNA, that is one strand of DNA, that is the 5' end, has a free phosphate group on one strand, whereas on the other strand the 3' end has the free hydroxyl group. And similarly, one strand of DNA has free hydroxyl group at the 3' end and the other has the phosphate group at the 5' end. Then we discussed that hydrogen bondings and base stacking interactions are responsible for its stability. Then we discussed that three consecutive base pairs on a DNA molecule constitute a codon. And then we discussed the central dogma, that is DNA first transcribes into mRNA and then mRNA transcribes into a protein. This is all for today. Thank you.